Welcome to Mycology Unmasked. In this episode, we are diving into the amazing cellular world of fungi, from what MBBS students must master to what MDs need for diagnosis, and finally, the cutting edge secrets uncovered by postdoctoral researchers. Stay till the end of the real world clinical tips, antifungal targets, and what researchers are discovering in 2025. So, coming to the clinical cell, let's begin with the very basics every MBBS student needs to know. So, the fungal cell wall, it is the rigid, multi-layered cell wall made up of chitin, beta-glucans, and manoproteins. These are the major components, and these are the target for antifungals. These are the target for antifungals. Example, echinocandins inhibit beta-glucan synthesis. So what is the importance of knowing the structure, the component of the cell wall? To know the components of the cell wall so that we can manufacture the antifungals to target the components, to destroy them, to know them, to destroy them. Now this is the cell wall structure. These are the fungal PAMs, means pathogen associated molecular patterns. Mannan, outside inwards, mannan, beta 1,6 glucan, then beta 1,3 glucan, and then chitin. This is the phospholipid bilayer, and membrane proteins are embedded inside. Phospholipid bilayer is the component of the fungal cell membrane. And this is a picture of the pattern recognition receptors. But these are present inside our body, inside human body. Pattern recognition receptors can recognize specific fungal pathogen-associated membrane patterns. So when we get attacked by fungal cells, PRR means pattern recognition receptors recognize them and try to neutralize them. Now, next is the fungal cell membrane. As we have seen, it is composed of phospholipid bilayer. Human cell membrane is also made up of phospholipid bilayer. But what is the difference in human cell membrane? There is cholesterol, but in fungal cell, instead of cholesterol, there is ergosterol. So when we design antifungal drugs like azoles or amphotericin B, they can destroy the ergosterol, not the cholesterol. So human cell won't get destroyed by the antifungal drugs. Azoles like fluconazole inhibit ergosterol synthesis, and amphotericin B binds to ergosterol and causes membrane pores. Next, coming to the morphology. Next is the fungal morphology. It may exist as yeast form or mold form, or dimorphic fungi also may exist. Yeasts means unicellular, example Candida albicans, they multiply by budding, and mold means multicellular, they can form hyphae. Dimorphic fungi, they can switch forms, their forms yeast and molds based on temperature. Example of mold form is Aspergillus or Aspergillus. Example of dimorphic fungi, Histoplasma capsulatum. So, how can you remember? By the mnemonic mold in cold and yeast in the heat. Means in mold in mud, outside body, these dimorphic fungi exist in mold form, and inside body, they exist in yeast form. That means in heat at 37 degree centigrade temperature. Now, hyphal types, they may be septate or non-septate or aseptate. Septate hyphae, Aspergillus, and non-septate or aseptate hyphae, example, mucor. So actually, in mucor, the septae are arranged in such a long distance that they cannot be seen in microscope. It is said that there is nothing as aseptate hyphal types. But what we see in microscope, it is aseptate. This is seen in KOH mount under microscope. Now reproduction may be sexual, may be asexual. Sexual reproduction less common in clinical fungi, pathogenic fungi. Asexual reproduction means spores by reproduction by spores and by budding. Spore types can help in identification of fungi. As for example, conidia seen in Aspergillus. If we see conidia and if other criteria we can validate, then we can assume that it is Aspergillus, or we can diagnose that it is Aspergillus. Now, this is the KOH mount picture. This is the gram stain picture. This is LPCB mount, and this is India ink preparation, India ink staining. This is the KOH preparation. Those are the yeast-like cells, and these are the pseudohypha. And chlamydospore at the tip present, and blastospores also we can see. This is the lactophenol cotton blue staining or LPCB staining. Microscopically, this is the lactophenol cotton blue staining in which we can find a dematiaceous mold that causes chromoblastomycosis. This is basically of Cladosporium caryonae. And this is the India ink preparation of Cryptococcus neoformans showing the capsule. This is a negative staining. And inside those, we can see the budding cells. The daughter cells are there. And this is the gram stain picture of fungal cells. This halo is due to presence of capsule. Now, the fungal organelles. What are the organelles present inside the fungal cells? Same as eukaryotic cells, nucleus, mitochondria, Golgi apparatus, and closed mitosis in some fungi. That means nuclear envelope does not break. What are the important structures? Spitzenkorper corpuscles. This directs tip growth in molds. This is not a MBBS core topic, but it is good to know. And voronin bodies plug septal pores found in filamentous fungi. Clinical significance of fungi. Fungal virulence factors like adhesins, which help them attach to host. Enzymes, example proteases, to break the host cell wall or cell membrane. And biofilms to resist the antifungals. So this is to attach, first attach them or attack, then to invade, and then protect themselves, defense. Fungal infections, this may be superficial, maybe systemic, or maybe opportunistic fungal infections for immunocompromised patients like diabetic patients, HIV affected patients, seropositive patients, or for patients with organ transplantation. Superficial means dermatophytes, infections by dermatophytes. Systemic infections can be caused by histoplasma, cryptococcus, etc. And opportunistic by Candida, Aspergillus, and mucor. Microscopic findings for different types of fungi: Candida, budding yeast or pseudohyphae, and pseudohyphae, Aspergillus, septate hyphae, and acute angle branching. By these, we can identify those fungi in KOH mount also. Mucor, broad, non-septate hyphae, and right angle branching. So, septate, Aspergillus, mucor, non-septate. Aspergillus show acute angle branching, and mucor shows right angle branching. Cryptococcus, this is the encapsulated yeast. So, special stain, India ink stain or mucicarmine stain. This is a very special one. Now, for MD or postgraduate level students, you have to know some more finer details. So, fungal cell wall, diagnostic target and therapeutic target as well for us. This is composed of chitin, beta-1,3-glucans, manoproteins. PAMS means pathogen associated molecular patterns. This is recognized by PRR, pattern recognition receptors like dectin-1. And what are the laboratory relevances? 1,3-beta-D-glucan assay we can perform. This is a serum test for invasive fungal infections. And azole resistance like ERG11 mutation, efflux pumps also. Antifungal target like echinocandins, example, caspofungin. Antifungal azoles target structure is the ergosterol synthesis. Polyene, that is amphotericin B, target is the ergosterol itself. Inhibit ergosterol synthesis. Echinocandins, beta-glucan in cell wall. And griseofulvin target is the microtubules, that is it inhibits mitosis. Now cell membrane for cell, those were the antifungal targets in cell wall, and cell ergosterol is found in cell membrane. So this is a total antifungal target recap. Especially in cell membrane, contains ergosterol, azoles inhibit lanosterol 14 alpha D methylase. Polyenes bind directly to ergosterol, that is amphotericin B, and inhibit them. Resistance mechanism, ERG11 mutation, and efflux pumps, example CDR1 and MDR1 in Candida albicans. Those are the antifungal mechanisms. Now, a few more words about morphology and identification. Yeast versus mold versus dimorphic. You have to know hyphal structures like septate in Aspergillus, non-septate in Mucorales. Comparative study is much more important and effective also. And acute angle branching in Aspergillus, A for A, you can remember like this, and right angle for Mucorales. Important for LPCB mount, lactophenol cotton blue, KOH preparation, and tissue histology like GMS or PAS10. GMS or PAS10. GMS stains means this is a histological stain used primarily to identify fungi and fungal structure like hyphae, yeast, and spores by staining them black. The full form is Grocott's methenamine silver stain, and against a green background, black stain of spore, hyphae, yeast against a green background. This is the GMS stain, primarily for identifying fungus. And what is PAS stain? This is the periodic acid shift stain. This is also a special histology, mainly used in histology. It detects polysaccharides and other carbohydrate-rich molecules and stain them as magenta color. So this is important for glycogen storage disorders, diagnosis of glycogen storage disorders, and it also can identify the fungal cell walls. Now, reproduction and sporulation. From this, we will discuss in next video. So today, up till this, you learn and remember. And if you like the video, please don't forget to press the like button and also share and subscribe. Thank you. Have a good day.